pleasure to introduce one of our own colleagues, Swati Patel, from the Department of Math. Swati has been here for 3 years already, but I feel like we in IB don't know her well enough because of things, pandemic and being all the way up here. So I thought it would be a great opportunity to have her come on here to tell us stuff she does, because I think my sense already is that she's an underutilized resource for us biologists on all kinds of fronts, both the ecologists and the population geneticists and the evolutionary biologists. I think she kind of integrates a lot of what we care about and does it formally with mathematics. I don't know. Anyway, but the formal introduction. So I met Swati at UC Davis when she was doing her PhD with Sebastian Schreiber in applied math. She went from there after doing some really cool stuff on coexistence theory in the context of evolutionary dynamics. She went on to do a couple of postdocs, 3 postdocs in Vienna, at University of Warwick, and between that was Tulane. Still focusing on dynamical systems, but really from my perspective, in a unique way, bringing genetics, evolutionary genetics, into ecological dynamics. So that's in the context of just species interactions in general, community ecology, population biology, but also in the context of disease dynamics. And I think she's going to touch on a lot of those things today. We'll see. So that's what I do. Thank you for coming.
Yeah, thanks for inviting me. Yeah, it's really exciting to be here and get to interact with people in IB. I actually started off my first research experiences as an undergraduate working on phylogenetics problems at the Field Museum of Natural History, and it's kind of amazing to go to research and be around such great displays of natural history and life, and I'm getting to feel that vibe here again, being in this building a little bit. The mathematics department does not have that. So as many of us in this room, I started as a child with the fascination of the diversity of life forms that exist. And there's many interesting questions I think we can address about trying to understand the diversity of life, from macroevolutionary processes that result in these diverse life forms through speciation events and natural history and geological events. But what I work on now is more on shorter timescales and trying to understand, using mathematical modeling, what enables these species to persist over time in these communities that they live in. I use a dynamical systems approach to try to address this question. I do mathematical modeling of species interaction and analysis of those models to think about persistence of species. So what the main question I'll talk about today is what enables species to persist. So some early modeling, one more slide. So persistence, understanding the processes that lead to persistence has a variety of applications I'll talk about a little bit. So on one end, we might want to think about a species, why a species, or what enables a species to persist, to be able to have conservation efforts, such as those that exist for the Menzies wallflower, which is a species in California that's been subject to restoration efforts. On the other hand, we might want to understand what enables a species to persist in order to eliminate, for example, harmful species, those that cause diseases, such as the soil transmitted helminths, that I've drawn, that there's a picture of here, or the spotted winged Drosophila, which is an invasive species in Oregon that causes harm to the agricultural industry, and is the subject of some work done by a few people in our agriculture department here. So I'll touch on these 2 examples in the last part of my talk to talk about related to that. So mathematical modeling allows us to be able to study these kinds of systems and understand what enables their persistence. So some of the earliest work on understanding persistence comes from a classical example of 2 competing species. So this is a textbook example that one might be familiar with from a theoretical ecology class. So here is the model models two different species, their densities, through a differential equation system where you have the species densities and then a growth rate here that depends on these interactions. You have the interaction, intraspecific competition and the interspecific competition. And analysis, so this is a two-dimensional system, and turns out we can fully analyze in some sense the possibilities and classify the possibilities of the dynamics. So here what I'm drawing is generally the shape of the flow or the solutions of these differential equations here. And you see 4 different possibilities. In this first, you can see that species 1 will exclude species 2. Species 2 can exclude species 1. And then here you see an interesting pattern where it depends on the initial starting point of the population, whether species 1 excludes species 2 or species 2 excludes species 1. So this is labeled the priority effect because of that dependence on initial condition. And then finally, in this last case, you have this case of coexistence, right? Again, the flows here describing the pattern of the trajectories of the population densities. Interestingly, in this case, you can fully characterize this by what's happening on the boundary of the dynamics when roughly one species is very close to being extinct. And that which of these 4 possibilities results depends on the 4 parameters and their relationships. And so we can begin to understand what enables coexistence by looking at the requirements on the parameters that allow for that last case there. And it has a nice, so this is the condition on the parameters, which has a nice biological interpretation here. Says that 2 competing species will coexist if each more strongly limits members of its own species more than the other species. So this is a mechanism of coexistence from classical theory. And I highlight that this is a conclusion just under this model here. So this is 2 species. Of course, species rarely are just 2 species. They live in more complex communities. And in the 90s and the 2000s, there was a lot of work done to understand persistence of more complex ecological communities or trying to generalize these those kinds of conclusions. So a lot of the work that had existed until more recently focused on species interactions, but importantly, it ignored intraspecific variation. And there's lots of reasons why we might want to include intraspecific variation or different kinds of intraspecific variation that could be important to think about. So for example, one is that you might have variation in a genetically determined trait where now there's certain genotypes within the species and that affects importantly in an important way its interactions with other species. And then because of these interactions, that species may evolve over time and genetic components may change, and that can create this kind of so-called eco-evolutionary feedback. Other kinds of structure that can be important are these different life stages. This is kind of in my way here. Different life stages that interact differently with the different species that it interacts with, or sexes. So we may want to think about this kind of variation here. And a lot of my work is aimed at understanding that. So this is just one empirical example of how variation can be important for persistence. So this is a species of mustard. Trying to figure out the best place for that. The species for mustard, where this species has the ability to produce a toxin. So cinnagrine is a toxin that can help it compete against other species, other plant species. So in the presence of other plant species, the high cinnagrine or the toxic form can invade and become more prevalent in a plot. And then when these species go down, since the toxin is costly, it will the species will evolve to reduce the frequency of that toxin or the prevalence of that toxin, and the low synegrin morph will start to invade in a high synegrin plot. And then once we're in a low synegrin, once most of the plot is of the low synegrin form, then the other plant species can come back and invade. So it's an example of how intraspecific variation might enable persistence of different kinds of or more diverse forms. So the main question that I will talk about today through some models is when and how is internal variation and population structure important for persistence? And I will start by just giving a brief mathematical framework of how to think about, or one way we think about complex ecological interactions and accounting for variation within species. And that's really work with Sebastian Schreiber from my thesis, and then I will present two specific examples of two models. One of them is a genetically structured model, that's an eco-evolutionary model, and I'll talk about some of the interesting feedbacks between ecological interactions and the underlying genetics within one of the species. So this is an abstract model. And then finally, I will talk about some work motivated by the goals of eliminating a harmful species through this stage structured model. And that's this is work with Reinhard Berger, and then the last project is work with Patrick at OSU, Patrick Killing here, who maybe some of you know. Okay, good. So I'll start with just a general framework. I'm going to start, so I'm thinking about some number of species interacting, and I have xi as the density of species xi, sorry, the density of species I, not xi. And then each species has a per capita growth rate that depends on the densities of all the other species through these interactions. And we can write down a fairly general form of this system of equations right here. So these are just describing species interactions in an arbitrarily large community. And to this, what we do is add a second variable or a second set of variables that capture the structure or the internal variation within species. So for example, this could be the frequency of certain types within the population, or it could be the mean of some kind of trait distribution, right? And then I write down a general equation for that, how that changes over time. So this is the general structure of the model. So it's fairly general. I haven't really specified very much here, and that's purposefully that way. And what we do is develop some foundational theory that can be used as a tool to understand systems that do fall under this category or this form. So these are general equations, but they're arbitrarily large, right? So they're really high dimensional, they're potentially really high dimensional if you're talking about a lot of species or a lot of keeping track of a lot of frequencies within a species. And so it's not always feasible or useful to think about characterizing all possible dynamics, especially for more than 3 variables. So instead of trying to characterize all possible dynamics, we take like a coarser grained approach at trying to understand these equations. So on one end, we can look very locally, assuming there's some kind of equilibrium point. We can just look locally around that and see if you perturb the population densities in one way, or if you perturb the frequencies within the species in some way, does it go back to that equilibrium state? And on the other end, we can look really globally at this, and it's the mathematical notion of coexistence called persistence. So I'll describe what I mean by that. It's basically saying if you start with some initial population density, then over time, it stays in a region where it does not get close to 0. So it's bounded away from 0. It never gets, there's some lower bound to it staying above that lower bound for all of time, right? So I tried to draw that in this picture. And so we can actually characterize what conditions are needed on these equations in order to get persistence and stability. So this is providing some very specific conditions. Okay, it's kind of technical, but what I wanted to point out in words, what translating this to this technical condition is that what you need for persistence in this system is that if on average in time and averaging across all the species, the growth rates are positive, then that whole community persists. So you can kind of look on average in these two ways, averaging across species and averaging in time in some way. Then all of the species will persist, meaning they'll stay away from zero or stay away from getting close to zero. Yeah, I wanted to put this here because I'll use it in some specific systems here. So I'll move on to the next part of my talk, which is developing a genetically structured model, an eco-evolutionary model. And I'll apply some of the persistence idea from before here. Okay. So the genetically structured model that we worked on was looking just at a 3-species interaction. So this is it's called an apparent competition type interaction. You have 2 prey that share a predator. So these are pretty common in, for example, insects that live on 2 different plants can compete indirectly if they have a shared predator. So and the reason they call it apparent competition, if one of the prey species starts increasing density, that's going to lead the predator species to increase in density. And then that creates more pressure on the second prey here. So it seems like they're competing, even though they don't really directly interact. So in the apparent competition interaction, we assume that there's variation in this predator species, and the trait that is varying determines the consumption of each prey. So it consumes each prey at a different rate depending on some underlying genetic structure here. And so this is just an example of the rate of consumption depending on some genetic value gene. Then we assume that that genetic value depends on 2 diallelic loci in the genome of the predator here. So 2 loci, each with 2 possible alleles that are going to determine the genotypic value of each individual predator here. Okay, we purposely did 2 diallelic loci. I wanted to explore more complex genetic architecture, but still be able to have it be analytically possible to study the system here. So we can write down, so some assumptions. So I'm going to assume that one of these loci has a bigger effect than the other on the genotype. And in the process, the selection on one locus is going to induce a selection on the other locus just because they are potentially linked in the genome. So there's some kind of linkage disequilibrium, or in other words, some statistical association of the two alleles at the 2 loci. And we know that, and we also model that recombination within the individual predators can break down this kind of linkage that is generated from selection processes. So we can write down a set of equations here. So on this side, these are the equations for the dynamics of the different species. And then here I have P, and P is the frequency of the one allele at one locus. Q is the frequency of the one allele at the other locus. And D is that linkage disequilibrium, or the statistical association of the 2 alleles here. So in analyzing, we can analyze these equations to try to understand what enables, in particular, persistence of species, and in addition, what enables internal variation to be maintained within the predator species. Okay, so I'll start with just describing some results for the unstructured population. So this is just the left-hand side equations of just describing this ecological interaction. And those say that for sufficiently large carrying capacities, one prey will exclude the other prey, just as in the two species competition model, where you have the cases of exclusion, one species excluding the other. But in the structured population, we see that there is persistence here. So for sufficiently large carrying capacities, all three species will persist. And interestingly, the locus one with the larger effect on the trait remains polymorphic. So the one with the loci in the predator species with larger effect on the trait remains to stay variable within the population, right? For this result, we really are just applying the theorem from the first part of my talk. So this begs the question, well, what happens to locus 2? And what happens to locus 2 really depends on the recombination rate within the predator, right? In some cases, it will also remain polymorphic, but in others, it will get fixed to one allele or the other. So this result is about persistence in the population, and it just erased that. And to examine what happens to the other locus, we look at a more local property of stability. So there is some equilibrium point here that we can identify where you have a polymorphism in both loci. But this is 6 variables, right? So understanding stability of in a 6-dimensional system is non-trivial. It's not really tractable generally. But what we do is a change of variables where we have the total prey density and then the difference between the two prey densities. And that allows us nicely to decouple the dynamics, at least locally, into the dynamics between the difference between the two prey and the allele frequencies and the total prey density, total predator density, and the linkage equilibrium. So this is nice. It's cool because it helps us identify some key biological feedbacks here. So decoupling into 2 sets of separate equations. And it also enables us for it to be analytically tractable. So I'll share some numerical results at least. So for a sufficiently small r, that's that recombination rate. So if recombination rate is really small, then the equilibrium is stable. So you have this maintenance of the polymorphism here, P and Q, and the population densities. On the other hand, if recombination is large, then the equilibrium is unstable, and you can see that there's fixation of the allele at the locus with lower effect on the trait. So what happens at intermediate recombination rates? Well, you get these interesting cycles here. You get cycles where the allele frequencies stay fixed within the predator species, but you get cycles between the total predator or total prey density, the total predator density. So those are kind of like the classical predator-prey dynamics. But interestingly, you get this cycle with the linkage that's happening within the predator species. So there's a cycle between linkage of alleles and the total prey and predator densities here. And I'll try to describe that in a few slides where an explanation for why these things cycle. So as I said, we can understand the kinds of feedbacks that are happening from the decoupling that we did. So this is a feedback that is that we called alternating direction of selection feedback. So here we start with, let's say the difference between the two prey goes down. Then there's the selection gradient changes to one direction. So this creates a selective pressure on the predator, which drives the allele frequencies to go down. And then allele frequencies going down reduces pressure on the two, one of the prey versus the other, and that should be X minus. X minus goes back up, and you have this kind of cycle that results from there. This is very akin to the type of cycle that we were talking about that led to persistence of the this species of mustard with the other plant species, right? You're alternating a selection direction, and that's allowing for this other species to persist here. So what's happening with this other feedback here? So this one I think is maybe more interesting. We call this the fluctuating strength of selection feedback. So this is a feedback between the total prey, the total predator, and then the linkage within the predator. So here, if the total prey species goes up, what that does here is leads to a sharper selection gradient here. So with the sharper selection gradient, if you have stronger selection, it can lead to more linkage disequilibrium. It kind of combats recombination. So that allows, it decreases the linkage disequilibrium, which is increasing it in magnitude because it's negative. So it decreases the linkage disequilibrium because you have stronger selection. The decreasing of the linkage disequilibrium puts pressure on all of the prey species, because in some sense it's making the predator species stronger, which then completes the cycle by decreasing the selection gradient, which allows for an increase in the linkage. So this is an explanation for those other kinds of cycles that we were seeing there. Okay, so I'll just conclude this part of my talk. So some takeaways, some biological takeaways from this analysis. The evolution of the predator enables the coexistence of these two prey, and that's really through this alternating the direction of selection kind of feedback. Additionally, our first result on persistence says that the locus with greater effect on the trait is the one that remains polymorphic. And then recombination within the predator may generate cycles between the prey, predator, and the linkage of alleles within the predator. So this is an example of how you can have interesting feedbacks between the genetic composition within one species and then its species interaction. Okay. Good. So in the last part of my talk, I'll talk about a model of a life stage structured model of a single species at a time. So this is work motivated by two examples of systems that I've been thinking about. And they're the ones that I presented at the beginning of my talk. So one of them is these soil transmitted helminths. So these are parasitic worms that live inside guts of humans as well as other animals that are important for agriculture in particular. There's a number of different species of them, but they all share an overall structure of their life cycle depicted here. So they spend part of their life cycle in the soil, and in the soil, they transition from eggs to larvae. And then at this stage, they get taken up by a host through contamination or contact. And they get taken up into the host, and within the host, they'll transition from larvae to adults. Here, let me get a drink of water. So these are worms that cause disease within their host. And so they've been the subject of different kinds of control efforts to protect populations against them. And one of those control efforts is known as massive drug administration. So this is where different public health organizations will go out to communities for humans where this disease is thought to be prevalent and just massively try to treat a big subset of that population at one time. And they try to do this periodically, maybe once or twice, in some cases, 3 times a year. Here. Yeah. So the second example is the second motivating example for this next work is the spotted winged Drosophila, which is an invasive species that infects agriculture in Oregon, amongst many other places. It has a life cycle structured into different classes based off of the life cycle of this species. And similarly to the helminth populations, they're also under active control efforts through insecticide spray. So especially on farms, they may spray periodically a few times a year to try to control this species here. Okay. So these are examples that motivate our model. And in our model, we address questions, some natural questions that we may want to address, which are how often should we treat to effectively control this harmful population, accounting for this structure, and how does the population structure and the differential impact of treatment on each different class affect how often we should treat. So importantly, in the previous slide, I should have mentioned that in this case, the treatment or the control effort only affects really adult worms because you're only doing it within the host population. And in the insecticide spray, the spray has a different effect depending on the life stage. So it will kill some proportion, and that proportion depends on which stage of life that you're talking about. So how does this structure impact how often we should treat? So I'll abstract from those 2 specific examples and to build a model. So here I have a population structured into 2 distinct classes. And there's movement between and out of the classes that I'll capture by a matrix A. So here is the densities of the 2 classes, and then the matrix A that's describing the movement between and then out of the 2 classes. On top of this, there's some kind of pulse control intervention that might impact each class differently. So in this picture, I've only drawn it impacting one of the 2 classes. And this is captured in this matrix D, a diagonal matrix. It has it's basically saying some proportion of class 1 and class 2 will be removed from the population due to the control. And this happens periodically in time, given by tau. Okay. So we can write a pretty simple model to describe this. So here's the dynamics of the population with in between treatment pulses. So this is the dynamics every time you're not at any integer multiplication of the period here. And then at the time of the control intervention, it instantaneously changes the state of the population from one point to another point here. So that's captured in this equation here. So this is our model. And we can understand some fundamental qualitative behaviors of this model to tell us how often we should treat. So an assumption I'm going to make is that the eigenvalue of A is greater than 0. If you don't know what that means, you can just interpret it as the population is going to grow in the absence of these interventions. So I'm assuming the population is growing if I don't intervene. And then from this model, there's an object called the spectral radius of this resulting matrix. And what that does is it governs the long-term dynamics here. Okay. And so we really want to understand the behavior of this as a function of tau to understand how often we should treat, because that's what's telling us about the long-term dynamics. In particular, there's two different cases. So if that mathematical quantity is less than 1, then 0 is stable. So it's driving that population to 0 or elimination. But if that quantity is greater than 1, then this control is ineffective, 0 is unstable, and the population is going to continue to grow despite any kind of control that we're doing that reduces the population. So here, the timing will affect whether we're in case 1 or case 2. Okay, and additionally, we might want to minimize that the spectral radius because it implies more efficient control. So the smaller it is, the better for control, roughly speaking. Okay, so we want to understand the behavior of this. So mathematically, again, it's another technical result, but I'll describe the implications of it. So what we do is show that the behavior of this map with respect to tau is convex. But what that means will become apparent in the following slides. I want to juxtapose this again by thinking about the unstructured population first. So if the population is unstructured, we have a dynamic described here. And you have this one behavior for the spectral radius. So that in this case, that quantity is always increasing with tau. And so what you can interpret that as saying you want to treat as often as possible. That's the best strategy for getting closest to elimination, treating as often as possible. This is unstructured. So what happens when we structure the population? Well, so for this, I want to identify the least controlled class. So I'm going to call that class K. That's the one where the treatment effect has the least effect on that population. And then I can divide it up into two cases, the possibilities. So the first case, if the least controlled class is self-promoting, so that's it. This term means that it's self-promoting, then you want to treat as often as possible. And the shape of this curve looks just like the unstructured population case. On the other hand, if you look at the case where the least controlled class is self-limiting, then you get this other shape for the spectral radius. And here you can see that there's an optimal tau in the range of effective treatment that is the most efficient at eliminating this population. And so here treating too often actually becomes inefficient here. And that's largely due to the structure and the self-limitation. Okay. All right. So we took those theoretical results, and I looked at a very simple model of the life cycle of the soil transmitted helminths and took parameters from the literature about how fast their life cycle is from transitioning between these stages. And I looked at the spectral radius. So here the pulse only impacts the adult worms. And then I looked at the spectral radius of that matrix, and these are 3 different species of helminths. So they have different parameters associated with them. But you can see this pattern of there being this optimal time or period to treat to be the most efficient at reaching the elimination targets for these populations here. Okay, so yeah, in the last little bit, I'll talk about some ongoing work. So this result that we have on the convexity, which is fundamental to being able to determine these behaviors of the spectral radius. This, interestingly, because of this technical condition, does not apply to this model type in particular. So some ongoing work is to extend this result to include a more broader set of model types, in particular getting rid of some of these technical conditions so that we can apply it to the spotted wing Drosophila work. A second thing that I'm thinking or working on is based off of the soil transmitted helminths. One thing you'll notice is in this model and trying to optimize the period, I'm assuming that the impact of treatment is constant in time. So every time I apply treatment, it has the same exact effect on the populations of worms. But we know that there's a lot of evidence, especially in the veterinary literature, that these helminth species are evolving in drug resistance. And so some ongoing work is to build and analyze a stage and genetic structured model here with these pulsed disturbances to try to tease apart what kinds of strategies of control can we use to drive this population to elimination in spite of the evolution of resistance to this drug. And this is ongoing work with 2 other people. Yeah, that's all I wanted to say. So, but I just want to end with some overall conclusions. Interspecific variation in population structure matter for persistence of species. Genetic dynamics within one species can impact the whole persistence of a whole community. And stage structure can impact how often or the period of treatment needed in order to drive elimination for a target population that's harmful. At least in theory, right? So there we go. At least in theory, right? So one caveat to all this work is that it's all based off of abstract and theoretical models, and we need to know if this is actually true and happening in reality. And it reminds me of this quote. This quote always rings in my head as I do my research. It's from Oliver Sacks, not too long ago, 2013. One has seen grand theories rise only to be toppled by stubborn facts. Right? So this inspires me. One of my main research goals with this quote kind of ringing in my mind is to really connect these theoretical ideas from abstract models to reality. And I think there's 2 potential ways to move forward to do that. One is to actually to be able to test these theoretical predictions on either ecoevolutionary dynamics or the impact of pulsed disturbances in an experimental system. I think for that, we would need a system where we can track some kind of genetic frequencies and population densities simultaneously, or be able to manipulate these pulse and experimentally generate pulsed disturbances. And then the second way that I would love to build and work more on is to build more empirically driven models. I think one of the big things in a lot of the ecoevolutionary models is that we don't have a very accurate genetic basis of traits. And this is a direction I think would be super cool and interesting to go into, trying to actually couple real genetic architecture of traits with their ecological interactions. And so this is a research dream of mine. And if any of this speaks to you, I would love to talk more to you if you think of an experimental system that would be able to address these kinds of problems. Please reach out to me. So that's it. Thank you. Yeah, Cheyenne.
Quick question on the results for the components. When you saw that dip in their in the spectral radius, was that like connected to maybe earlier life stages? If you target those like egg or larval stages, you have like a better impact of treatment.
Yeah. So this is based off of the model. Well, for example, here you see it with the model with just 2, and you're only impacting one of the 2 species at a time, or one of the 2 life stages at a time. So in the Hellman's case, the least controlled class are the eggs and larvae, the ones in the soil, because they're not impacted at all. And that is what causes this dip. It's essentially like inefficient to treat so often because you haven't had any influx from that other uncontrolled part, right? So like the stuff in the humans has already dipped down. You need to wait until they can come in from the other side in order to treat again, or for it to be effective to treat again. Yeah.
Yeah, I'm so sorry if you went over this already also. With your model, it's accounting for the least controlled class to have no effect when you do some sort of control, is that?
No, I mean, generally it can be anything, everything is controlled.
Yeah.
And you just look at the one that's least affected.
Got it, okay.
That's the only one that's the one that's going to affect the shape between this shape or the other shape. Yeah. So yeah, they can be like 90, 50, and 10% affected, and you just want to look at the one that's only 10% affected.
Yeah.
Yeah. And there's Molly.
Yeah, thanks for a really engaging talk. I'm really curious about the middle part of your talk and like the result that you got about the larger effect locus.
Yeah.
That you modeled leading to a more stable polymorphism.
Yes.
So I guess I'm just curious in general, how easy or difficult would it be to change that parameter, either that parameter or the number of loci that you could incorporate into that? Like, could you model the effect of the locus size or the impact of the locus or the number of loci?
Yeah. So, I mean, maybe this isn't exactly what you're saying, but I am like wanting to look at like 3 loci, 4 loci, and so on, and try to build a more general theory and see how that statement holds if you have a model with 3 loci or more. And yeah, I haven't done it yet. And that kind of addresses your question, but I guess the other way is to actually model like the number of loci involved, and maybe are you suggesting like that's evolving in time?
Oh, I just think it would be really interesting they're 2 totally separate parameters, but I think they're both really interesting, both the number of loci and the effect of any one of those loci.
Yeah, okay. Yeah, I mean, I did look at like the effect of loci versus the recombination. Let me see if I have it. I don't. But yeah, I can look at how much bigger is the effect of that one loci versus the recombination rate. And I did look at that, and it does affect like the overall dynamics, but the general pattern, like the possible dynamics are still like the same. So you just see like a larger parameter range, where if the loci effect is even larger, then you might see more cycling, something like that. Yeah. I'll see if I can pull it up. I have it in one of my older talks.
Or we can chat later.
Yeah. But I thought that was interesting because what I wanted to know is, well, is this true in reality? Do we see large, and maybe you know more about it, but do we see larger effect loci being more polymorphic in real systems? I don't know. Yeah.
So I know you said that it's like a theory and stuff, but for like the worms, is it too much to ask, like, did you find an answer in terms of like, when's the best day to treat them?
Yeah,
Like, where does that dip actually happen?
Yeah.
In their life history?
yes, let's see, you can probably see it in this, and I did look at it more carefully, now I don't remember. But I think it's, like this one is maybe like, the period is really low. It's like 10, maybe 15 days. This one's also pretty low, and then this one is the highest here. I do want to be careful about interpretation. Like, these are really simplified models. They're ignoring a lot. And so I wouldn't suggest that this should be interpreted in policy at all. Yeah, but the roundworm has the largest optimal, and I would have to look back at the differences in the life history parameters, but it has to do with how fast the cycle happens, and that the impact of the drug is different for all 3 species as well. Did I address your question?
Yeah.
Yeah, Mark?
Is there a relationship between that optimum and the period or the number of days where it flips from being negative to positive? And are those co-bearing or independent
Oh,
aspects?
yeah, I think they're independent. Like, I think you can probably produce a matrix A that has all kinds of patterns. I think, but that's left for further investigation. Yeah, so where it hits 1 is where anything below 1 would lead to elimination. And so is there a relationship between where it hits 1 and the optimal? And I don't, I think you can construct it all kinds of patterns. Yeah. Yes.
Yeah, thanks for this talk. In your species persistence model, it's a highly density dependent
Yes,
world.
yeah.
How much density independence can you put into this system and still get the similar result?
So you're talking about like the way general model
Yeah,
at the beginning, and it's just density dependence.
Yeah.
Yeah.
So if you put a little, like, how much density independence can you put into the system so that you have this 0 version?
What do you mean by density independence? Just like adding, if you were to throw some in.
Yeah, so let's say you have a stochastic improvement event or
Yeah.
a mortality term that comes to sweeps the
Uh-huh.
population. Things that we worry about a lot.
Yeah.
Is there a scalar so that this will work as long as the density independent processes are not bigger than X or Y.
Yeah. So I think the reason for doing it with just the density independence is a mathematical reason. Like, it makes us able to analyze persistence in the way that we're thinking about it. Because if you have a dynamic and you always have this like stochastic recruitment, it's coming in to the population, that's always going to kind of like maintain something there. It's not going to get arbitrarily close to 0 for all of the time.
And what about like mortality events that sweeps through?
Yeah.
It seems like it's really important to have this aversion to 0. As you approach 0, then there's a strong feedback so that your growth rate either enhances or your mortality rate declines very rapidly. What if you have the independent processes that really counteract that?
Yeah, I think this second model kind of gets that. You have these instantaneous events where you're having like a massive mortality. It's still density dependent though. Yeah, these are still density dependent, you're right. Yeah. I think that's another set of models that one could analyze. Yeah. Thank you. Yeah.
Thanks, buddy, one more time.
Well, thank you.